The next spider we need to discuss is Leptospira, and genus contain two main species, Leptospira interrogans and Leptospira biflexa. Any bacteria of the genus have microaerophilic type of respiration. It is of course spiral in shape, and if you will apply gram stain by nature of bacteria, it is gram negative. But as we said, bacteria, any spiral bacteria is poorly stained with the gram technique, and Leptospira is the mostly the more thin bacteria than other, and common staining techniques are not applied for that microorganism. So bacteria have unique shape. One or both ends of bacteria are hooked, and that hook can look in one or two different sites, and bacteria can resemble question mark or English C or S letters. And next slide, we will talk about again microscopic techniques to identify that bacteria. So microorganisms have two periplasmic flagella which are attached, connected to both poles of a microorganism. That's why it has so unique shape. And Leptospira interrogans is the main pathogenic species of Leptospira, and second one, Biflex, is a saprophytic species. It is widely distributed in water in different aquatic reservoirs and not associated with any human diseases. So Leptospira have very difficult antigenic structure, and actually it is classified into 22 serogroups and more than 200 different serotypes. And serotyping is important in epidemiologic studies, and actually different countries are endemic by different serotypes. So in usual medical practice, identification to species is enough and significant to put the diagnosis and initiate proper treatment. By nature, leptospirosis is a group of zoonotic diseases, and natural reservoir of bacteria is a wide range of animals, domestic and wild animals, including dogs and different type of rodents. Human for the disease is accidental host, but still can be ill. Usually, bacteria is delivered in nature with urine of animals, and it is highly distributed in soil and water, and can survive here for a long period up to 2 weeks. If you'll talk about identification of bacteria in microscope, we have the same absolutely techniques. It is a dark field microscopy, and you will have reflected light in the shape of spiral, long elongated spiral in the black background. We have different types of silver impregnation techniques. Bacteria will appear as dark blue or dark brown in the yellowish background. But both methods, the same as in the case with treponema, will not allow you to confirm that it is really Leptospira. They will show you only unique appearance of bacteria. And third method is immunofluorescence, which using specific antibodies labeled with fluorescent dye. That method is confirmatory microscopic method for Leptospira because antibodies are always very specific against precise antigen, and if they stick to bacteria, then you see the shape of microglobin in UV light. It is always Leptospira for sure. And if you use type-specific antibodies, you can identify even serogroup or serotype of microorganisms, not only the species of bacteria. As any other spiral bacteria, Leptospira is very difficult for cultivation. We have EMGH culture medium as the main medium for cultivation of Leptospira. It is named, it is actually an acronym from names of scientists who designed that culture medium, Ellinghausen and McCullough, and later it was modified, and nowadays we're using that modification by Johnson and Harris, and EMHG is the name of medium in the honor of those scientists. So culture medium is semi-synthetic with addition of rabbit serum, and bacteria can grow here native culture medium is transparent and have orange color, as you can see in the left part of the picture. And when bacteria grow in culture medium, it became non-transparent and darker in color, almost black in color. So the same problem as with other spirals, bacteria is very difficult and have complex structure of cell wall and structure of bacterial cell. And doubling time is about 12 hours up to 16 hours. It means that bacteria can multiply only twice per day. And of course, culture method is very long method for diagnosis, and usually it is not used in routine practice. So we have azole inoculation in susceptible animals or use of chick embryos, but not every lab is equipped with animal vivarium or have some conditions to cultivate bacteria in chick embryos. So usually diagnosis is done by some fast and rapid methods of identification of bacterial antibodies, but not with the classic biological methods, microbiological methods. So if we talk about diseases which can be caused by leptospirosis, usually we split them in two main groups. Any disease caused by leptospira with very mild duration is called as a leptospirosis. The same reservoir for bacteria as animals. Animals can have carrier state, means they not suffering, not have any symptoms, but they can spread bacteria in environment. And it is important if you will talk about domestic animals, because dogs who are kept at home, pet dogs, they can deliver bacteria with the urine. Of course, some bacteria will stick to the fur, and when owner of the animal will perform some cleaning procedure, even playing, some taking them on hands, they can get that bacteria in skin or conjunctiva. They can become infected even by contact with that animal. Of course, water and soil, which are contaminated with urine or some other biological tissues of animal, can be contagious, and disease can be transmitted by washing hands, swimming in aquatic reservoirs, or drinking water, or use of that water for other purposes. And agriculture workers, especially some rice pickers who have a prolonged and constant contact of hands with water, and some stones and branches are here, they can injure skin. Those people are mainly affected by leptospirosis. And actually, wide range of professions, mainly due to animals and wild nature, such as livestock workers, butchers, home domestic animal keepers, agriculture workers, and hunters, they are affected by leptospirosis in equal manner. But again, disease is endemic, and we have only certain regions, certain areas where that microorganism is prevalent. So in some countries, disease is not registered or registered very poorly. So of course, it's not the point to be afraid of any aquatic reservoir, because disease is not common in some countries. So bacteria needs some injury of the skin epithelium, or it can penetrate through intact mucus. Again, during swimming or playing in natural aquatic reservoirs, bacteria can get into mouth, nose, or eyes, and can pass through mucous membrane and invade into the blood. But in main number of cases, up to 90% of cases, bacteria will cause non-specific symptoms of very mild disease, resembles respiratory infection with headache, slight fever, muscle pain, weakness, but disease is self-limited, and spontaneous convalescence will be observed in few days after onset of the disease without any serious consequences. In 10 to 20% of cases, bacteria can cause a severe state, severe type of leptospirosis, which have different names. Common name is a Wales disease. Disease will manifest with acute appearance of vomiting, severe headache. In few days after infection, bacteria will spread from site of invasion to bloodstream and will be disseminated to multiple organs, including skin. And further duration is usually divided into three clinical symptoms or according to which organ or system of organs is damaged, we have different forms of the disease. Most frequent one is hepatorenal damage and heart damage. Hepatorenal form is as well known as icterohemorrhagic fever because of three main symptoms. Person will have yellowish color of skin, yellow conjunctiva, yellow mucus, means that jaundice will appear, and uh, damage of blood capillaries, especially skin capillaries, will lead to appearance of hemorrhagic rashes, as you can see on pictures provided in the lower part of the slide. Rashes have unique appearance. It is red or dark red in color, have and sometimes looking like broken capillary with multiple branches, and even conjunctiva can be affected, and hemorrhages in conjunctiva are observed. Of course, disease manifests with high fever and three main symptoms. Uh, uh, Yellow color of skin, hemorrhagic rash on skin, and high fever gave name of that form, icterohemorrhagic fever. Besides that, disease is manifested by multiple signs of renal and liver damage, such as severe edema of internal organs, of soft tissues, exchange of metabolism, presence of proteins in urine, accumulation of creatinine and some other factors in blood when kidneys are affected. Blockage of kidney capillaries will lead to renal failure with decreased urination, and damage of heart will lead to tachycardia or shortness of breath and common symptoms of uh, multiple organ damage, such as weight loss, severe fatigue, and other damages will be observed in those people. Second form of disease is central neural damage or neural form, where disease will manifest initially with symptoms of meningitis without any sign of bacterial or viral infection. We call it aseptic meningitis. All symptoms of common meningitis will be observed, such as extremely high fever, severe nausea not released by vomiting, rigidity of neck muscles, pain in muscles, afraidness of loud sounds, of bright light, abnormal behavior, even seizures and coma state can be observed in that type of form. But if you perform examination of uh, liquor, cerebrospinal fluid, symptoms of bacterial inflammation will not be observed. And third form is lung damage or lung form. It will manifest with abnormal functions of lungs, severe decreased blood oxygenation, high fever, dyspnea, abnormal type of respiration, and in severe cases, coughing with the blood will be observed in those people. So mainly, disease is dangerous because of complications. A massive damage of some organ can lead to organ failure. According to which organ is affected, different symptoms and different complications are possible. We can have even multiple organ failures, and especially in hepatorenal form, massive internal bleedings can be observed, and mortality rate can be quite high without proper treatment. So to put the diagnosis, you need to take specimen from the subject, and you should understand that initially about a week or 10 days of the disease are caused by presence of bacteria in the blood. And to confirm the diagnosis, you always need to take blood in acute stage of the disease. In later stages, bacteria will hide in different organs and tissues, and in blood it will be not possible to find it. Of course, you still can look for antibodies in blood, but bacteria will accumulate in multiple organs, including kidneys. And due to broken kidney capillaries, bacteria can pass through membrane and through channels, and it is present in urine. So that's how it is delivered in nature with urine, and actually diagnosis should be done by studying of the urine. If you talk about preventive measures, we had some vaccines, but in main countries, in main number of countries, they are not available, they are prohibited or not approved, and only limited number of countries have human vaccines. The main measure of prevention in multiple and main number of countries are non-specific measures and vaccination of animals. So the idea if animal is healthy, human cannot be infected. Of course, it is possible to do if animal is domestic, but if you talk about wild nature and some professions due to wild nature, vaccination of wild animals are not really possible, and then only non-specific prophylaxis is available. So the main idea is bacteria present in water. And if you like to hunt into camping or just some rest in wild nature, be sure you're not using infected water. If you see any traces of animal feces, or animal steps near aquatic reservoir, it's better to avoid any contact with that water. Even washing and cleaning hands some can lead to infection. Of course, if you need to drink water, it should be disinfect it in a proper manner, use some filters or boiling to prevent presence of bacteria in that water. If you have some trauma injuries on skin, it's better to close them with some bandage resistant to water, not to allow bacteria to penetrate in that wound. So in some countries, including Russia, we do have human vaccines, but usually amount of that vaccine is very limited and immunity after vaccination is short. So vaccination is indicated for laboratory workers who deal with that bacteria continuously and for some professions in endemic areas, but vaccination should be done several times continuously until a contact is identified. So in the left picture, it is Russian inactivated vaccine, contain 4 serotypes. Vaccine is for subcutaneous injection and again used in endemic area. Usually it is some eastern regions, Kazakhstan and some areas close to Asia. In diagnosis, especially in classes, we're using the erythrocyte leptospiral diagnosticum, which contains antigen of Leptospira and used for reaction of agglutination to identify leptospiral antibodies. But of course, that test is nowadays kind of old. It is ELISA test, microwell, 96 microwell plate with pre-absorbed antigens of Leptospira. After several steps of preparation, you need to add specimen from human body, and if that antigen or if antibodies against Leptospira are present, they will form a complex, and color of solution in well will be changed. So that test is for IgG, but as well we have IgM test and combined test. Principle is the same. Test consume more time than rapid tests. It requires for several hours, and again, it should be performed in laboratory. For fast diagnosis near the bed of the patient, we have the same rapid immune chromatography tests. Principle is again common for any test, and usually tests combine identification of both classes of antibodies, G and M. We talked already in previous lecture. It is a membrane filter paper in plastic case. We have window for addition of sample. In the case of Leptospira, again, it is mainly human serum, and that is for identification of antibodies. So only serum can be used, and one result window when we have 3 possible results, one strip as a control, one strip for antibody class G, and one strip for antibody class M. So you need to add serum sample, mix it with several solutions which are going with the set, and they will diffuse by that filter paper strip. And 3 membranes here are present. IgM is the most heavy immunoglobulin and the largest immunoglobulin, have the biggest size. So first membrane have more wide pores and channels. They will hold only IgM, but will pass IgG and control reagent. So second strip is for IgG. They have a smaller size, and the control strip is the last. So after addition of your serum sample and mix with different solutions, we can have different results. If only C strip is present, it means that control is working, test is representative. If C strip is absent, control not working, it means the test is performed incorrectly, or maybe some reagent expired, that test cannot be evaluated. So C strip always should give you result. And then you can have nothing more. It means that subject is healthy and no reaction is present. Subject is not immune, not infected, and you need to look for some other disease, or maybe stage is so early that immune response is not active still. Then you need to repeat that test a little later. And of course, you should remember that antibodies will be produced in about 5 to 7 days after onset of the disease. And if subject is ill for first or few days, the test should not be used at all. But later, with the progress of the disease, of course, first class of antibodies will appear, it is IgM, and if you have in that test two strips positive, C and M, it means that control is working, test is good, it can be evaluated. And you identified IgM in your subject, it means that disease is in early stage, and it is only the beginning, it is some first week or first 10 days of the disease. If you have only IgG and control strips, it means that subject don't have M class of antibodies, and disease may be in late stage, it is closer to convalescence. Maybe it is reinfection when secondary immune response will immediately will be activated with class G of antibodies. Or maybe it is immunity after previous vaccination or previous disease, if titer of antibodies will be stable if you will perform that test. And if you have all three strips positive, it means that IgM still present and IgG already appeared. It is a peak of infection. Disease is not an early stage, it is in the middle in average stage, or maybe it is start of decrease of symptoms, and IgM will decrease, IgG will increase with the time. So again, it is a diagnosis, and subject require medical attention and proper therapy.